Hello, this is Dr. Hanaa Assil, and today we're talking about paper chromatography. So this is mainly for the IGCSE chemistry, but it could be basics for advanced level or any chapter on chromatography. So let us take a look at what is paper chromatography and how do we use it. First of all, paper chromatography is a technique that we use to determine the number of components in a certain mixture. So I have a sample of dye or a sample of ink. I want to know how many colors are inside ink. Or I have a sample of amino acids and I want to identify the components to know which substance is present in this mixture. So for example, I have a sample of green ink. I want to know this green color is made up of how many colors, how many components. So I can put a spot of the sample at the bottom of a rectangular filter paper, which is used as a chromatography paper. And I dip the end of the paper into a solvent, for example, water or ethanol. Now, this solvent will move up the paper. It will soak up the filter paper or soak up the chromatography paper. And as it goes up, you'll find that it will separate the colors in the sample that we have into different colors. So, for example, it was maybe green at the beginning, but actually this green color is made up of what? Gray, green, yellow, red, blue. So all of these were mixed up in that sample. We separated them using paper chromatography. So if I have a certain sample of ink or a certain sample of dye, then I want to know how many different colors. So I can put the spot of the sample. So the first thing, if he says describe what you're doing, then we say put a spot of the sample on a baseline. If you see, there is a line that we drew near the bottom of the filter paper or the chromatography paper. This is called the baseline. And this is where I'm going to put a spot of my sample. So put a spot of the sample on a baseline near the bottom of a rectangular chromatography paper. And then we said place the paper in a beaker containing a very small amount of solvent. The solvent could either be, we said, water or ethanol. And notice that the edge of the paper is touching the solvent, but I did not put the sample with the baseline inside the solvent. It's just the edge of the paper touching the solvent, and we leave it and allow the solvent to move up the paper. One thing you need to realize is when I am trying to put a spot of the sample on the paper, I need to put it on, we said, something called the baseline, or we refer to it as origin line. Now, this baseline has to be drawn in pencil. This is because pencil or graphite in the pencil does not dissolve, and it will not go up and interfere with the experiment. If I draw the baseline in ink, then the ink will go up with the colors and the experiments will be ruined. Now, we also said that the baseline that contains the spot of the sample must be above the level of the solvent. I don't dip the paper into the solvent. I put only the edge of the paper so that the baseline and the spot that I put are not inside the solvent. This is to avoid dissolving the spot in the solvent. If I dip it all into the water, then everything will dissolve in the water and the water will become colored instead of having the process of chromatography happening. Now, when the solvent moves up the paper, we said it separates the sample into different spots. Why do they separate? So if I put a sample on my baseline and then I found that one spot, which is green in this case, went up very high, another spot did not go up that high, and another spot did not move up at all. Now, what is the difference between them? They are separated based on solubility in the solvent. The spot or the color that moves up more, that means it is more soluble in the solvent. The one that does not move up that high, then it is less soluble in the solvent. If a spot remains on the baseline and does not move up at all, we say this is because it is insoluble in the solvent. So the component that is more soluble moves up the paper faster. The component that does not move at all is not soluble in the solvent. When the same substances are allowed to move up the paper, they will go up the same height. So for example, this was a chromatography experiment, and we found that, for example, we had the yellow spots went up the same height, so these are the same substances. The light blue spots went up the same height, so these are the same substances. So when we have several spots next to each other, if they go up the same height, that means they are the same substance. And we say they have the same RF value. What does RF value mean? RF value means the distance moved by the spot divided by the distance moved by the solvent. So we put this paper, for example, into the beaker and we allow the solvent to move up. Now I want to determine the RF value of the red spot compound 3. Can you see that? I want to determine the RF value of substance 3, which is the red spot on the right. How do I determine RF value? I get a ruler. So we need a ruler to measure the distance from the baseline to the spot divided by the distance from the baseline to where the solvent went up. Where the solvent went up is called the solvent front. So the distance moved by the spot divided by the distance moved by the solvent is called the RF value of the substance. Now each substance has a specific RF value. So if I put a substance and I did the RF value of this and chromatography of this, and I found these spots going up, you can see that the RF value is actually an indication of how far the spot went up. So the blue spot, for example, we calculated its RF value. You remember how to calculate RF value? The distance moved by the spot over the distance moved by the solvent. So ruler from the baseline to the spot divided by from the baseline to the solvent front. So that blue spot, if you sit down and calculate it, it is RF value is 0.4. But even if without sitting down to calculate it, RF value 0.4, that means it is less than halfway up. So if the spot is less than halfway up, then its RF value is less than 0.5. If it is exactly halfway up, it is 0.5. If it is a little bit higher than halfway up, then the RF value, for example, for this brown spot is 0.65. If the spot goes up a lot near to the solvent front, then it will have a higher RF value of about 0.8 or 0.7. Now, remember that the highest RF value you can have is 1. And that is if the spot went up to the solvent front. Then the distance traveled by the spot over the distance traveled by the solvent is 1. So remember that RF values of any spot are usually, or they have to be, less than 1. Okay, what if we're doing chromatography of spots that are not colored? For example, sugars, glucose, fructose, these are not colored. Amino acids are not colored. So if we do chromatography of sugars or chromatography of amino acids, the spots are not colored, so I don't see anything. So this is after I've done my chromatography, but I'm not seeing anything because the spots are not colored. So in that case, we take the chromatography paper after we finish and we spray it with a substance that gives the spots color and make it visible. This substance is called a locating agent. So, for example, these substances, sugars or amino acids, are not colored. I take the chromatography paper after we do the experiment, spray it with some sort of substance which we call a locating agent, and we find the spots appearing. So why are we spraying it with locating agent? To give the spots color, and this is another investigation. He's saying an orange drink may contain artificial colors E110, sunset yellow, or E129, Allura red. These are two substances. I don't care exactly what they are, but they're two substances, and we want to plan an investigation to determine the presence of these artificial colors in a sample of orange drink. So I have a sample of orange drink. I want to know, does it have E110 or not? And does it have E129 or not? Again, what do? We said, put a spot of the orange drink next to a spot of the E110 and the E129 on a baseline near the bottom of a rectangular filter paper. Put the paper into a beaker containing a very small amount of water. Now, when do we say water and when do we say ethanol? We say water if I'm talking about food colorings or dyes or ink. Then in that case, my solvent is water. We use ethanol if he tells me that the substance is not soluble in water. Then I say I'm going to put it into a very small amount of ethanol below the baseline. Allow the water to move up the paper. Now, if the orange drink has spots the same height as the artificial colorings, they are present. So in this case, does the drink have E110 or E129? It has E110. Since the E110 went up the same height, the spot from E110 went up the same height as the spot from the orange drink. Again, if he says a student produces this chromatogram for 4 dyes, so he has A, B, C, D, and he wants to know which of them is blue and which is red and which is yellow. And the question is, which one of the dyes contains 3 colors? Remember that a dye goes up vertically or on the same line going up, and he wants to know which one of the dyes has 3 colors. It is the one that separates into 3 spots. So the one that has 3 spots, this is the one that has 3 colors, so that is D. Which one of the dyes contains 1 color only? If it has 1 color only, that means it gave only 1 spot. So that is A. Which statement about paper chromatography is correct? He's saying a solvent is needed to dissolve the paper. Do we use the solvent to dissolve the paper? That's wrong. We use the solvent to dissolve the components of the sample. Paper chromatography separates mixtures of solvents. Is that what we do in paper chromatography? No, of course not. We separate the mixtures in a sample or the components of a sample. The solvent should cover the baseline? No, we said the solvent should be below the baseline. The baseline should be drawn in pencil? Yes, we said the baseline should be drawn in pencil. Then here he's saying amino acids are colorless and can be separated and identified by chromatography. What additional apparatus is required to identify the amino acids present in a mixture? What did we say about amino acids? We said actually they're not colored. So I will not see anything on the paper unless if I use what? Unless if I use a locating agent. And also, in order to identify them, I need to know their RF values. So the RF value, that means I need a ruler. Okay, so that means that I will need a ruler to determine the RF value, and I will need a locating agent to give the spots color to make it visible. Please note that during the experiment, he's covering the beaker with a glass cover. Can you see that? The glass cover is there. Why? To prevent evaporation of the solvent. I don't want my ethanol, for example, if I'm using ethanol, we don't want it to evaporate. So the glass cover on top of the beaker is to prevent evaporation of the solvent. A sample of green food coloring was separated into its component colors using paper chromatography. The results obtained are shown. So he got a blue spot and a yellow spot. And he's asking what is the RF value of the blue spot. He said, how do we determine RF value? We said RF value is the distance traveled by the spot over the distance traveled by the solvent. And that means in order to determine the RF value, we measure from the baseline to the spot. He wants the blue spot. From the baseline to the spot, divided by from the baseline to the solvent front. If you do that with a ruler, you will find that it is 0.45. And without even doing this calculation, you can see that the blue spot is almost in the middle of the paper, and that means its RF value will be somewhere around 0.4 or 0.5. Chromatography is done on a mixture containing a drug. The drug has RF value of 0.66. Which spot on the diagram or on the chromatogram represents the drug? So he's saying which of these spots has an RF value of 0.66. Now, without even sitting down and doing any calculations, obviously, if something has a spot of 0.66, that means it is slightly above the middle. So it cannot be C, it cannot be D, it's probably B. And if we do the calculation, he's saying the distance for B is 9.9, and the distance for the solvent front is 15, and that means that the RF value is 0.66. Do we understand that? Okay, this question says the diagram shows the chromatogram obtained when 4 samples of amino acids were analyzed. The paper was sprayed with ninhydrin. So when he says the paper was sprayed with something, even if you don't know what ninhydrin is, then you should know that if we spray a paper with something, then that something is a locating agent, and we sprayed the paper with it to give the spots color. Because here he's talking about amino acids, and we said amino acids originally don't have a color. So we have to spray it with a locating agent in order to give it color. So the question here says which amino acids could possibly be the same. Again, how do I know if something is the same as another one? If it has the same height, if it has spots going up the same height. So the ones that are the same here are samples 1 and 3. And then he's saying which amino acid sample contains more than one amino acid. So you can see that substance 1 gave 1 spot, substance 2 gave 1 spot, 3 gave 1 spot, but 4 gave 2 spots. And that means it has more than one component or more than one amino acid. A chromatography experiment was done to separate a mixture of 4 substances. And he's saying the RF values for these substances are 0.3, 0.5, 0.8, and 0.8. So he has 2 spots having the same RF value. And then I have another one that is 0.5. The one that is 0.5 should be somewhere in the middle of the paper. And another one that is 0.3, so that should be towards the bottom of the paper. So which of these has the substances he's talking about? You have to remember that if spots have the same RF value, they will appear one spot on the chromatography paper. They will be on top of each other. not next to each other. So my answer is C, not B and not D. The substances in B, the two spots on top of each other, will have different RF values. D, the spots that have the same RF value do not separate into two spots. They have the same RF value, so they will be on top of each other. That means they will cover each other. So my chromatogram will be C. A student used paper chromatography to separate a mixture of colored dyes. The diagram shows the apparatus. Draw a line on the diagram to show the level of the solvent. So he's showing me where the baseline is, where he put the dye, the spot of dye, where will be the level of the solvent. We said the level of the solvent has to be below the baseline. Then the question says, suggest a suitable solvent that could be used. We said, which solvent should we use? If he's talking about dyes, we said dyes or food colorings or ink, then my solvent should be water. If he says up in the question that the substance we're talking about is insoluble in water, then we say we're going to use ethanol. What could be used to put the mixture of colored dyes onto the paper? How do I put the spot of the sample on the paper? We just use a drop to put a drop on the basin. Then he says the clips hold the paper in position. Why is this important for the chromatography experiment? Why is it that I should keep the paper straight up? Because I don't want the paper to slip down into the solvent, so I want to keep the mixture above the solvent. The diagram shows the chromatogram obtained from 4 dyes, A, B, C, D. Give one conclusion that can be drawn about dye B. So, what can you say about dye B? Well, first of all, dye B has a spot the same height as D. So, B has D in it. Or you could say, B gave me 2 spots. That means it contains 2 substances. Suggest why dye C remained on the baseline. Can you see C? The spots stayed on the baseline, did not go up. Remember we said these spots separate based on what? They separate based on differences in solubility in the solvent. So if a spot does not go up at all, what does that tell you? It is insoluble in the solvent. It doesn't dissolve at all, so it stays there. RF values are used to identify the compounds. Calculate the RF value of dye A. Can you see dye A? How do we calculate RF value? Here he is telling me that it is the distance traveled by the spot divided by the distance traveled by the solvent. So in that case, you bring a ruler and you measure from the baseline to the spot of A divided by from the baseline to the solvent front, and this comes out to be at 0.6, and that is reasonable because spot A is a little bit just above the middle of the paper. The diagram shows paper chromatograms of 4 substances. Which 2 substances are pure? How do we know if something is pure? Of course, if it is pure, that means it has only 1 substance in it. That means if I do chromatography, it should give me only 1 spot. So which of these gives only 1 spot? Obviously, it is X and Z. The diagram shows a chromatogram obtained from 3 sweets, X, Y, and Z. And he's saying how many different red dyes are present in the sweets. How many different red dyes? Well, he's giving me the colors, red and yellow, yellow and red, and so on. How many different red dyes? Well, these red dyes, if they are the same height or the same RF value, that means they're the same substance. So the 2 spots on top are the same. The two red spots on top are the same, and the two red spots at the bottom are the same. So actually I have only two different red dyes. This question says a colored dye is separated by chromatography. One component of the dye moves a distance of 13 centimeters. So this is the distance moved by the spot, and has an RF value of 0.86. So that's the RF value. Which distance did the solvent front move? So this is the opposite. We know that the RF value is what? The RF value is the distance traveled by the spot over the distance traveled by the solvent. And then we said he gives me what? He gives me the RF value is 0.86. The distance traveled by the spot is 13. And we're required to get the distance traveled by the solvent. So, of course, just solving from the equation, the D comes out to be 15.1. So this is 15.1 centimeter. Okay, and this is the end of our video on paper chromatography. I hope this was clear and easy to understand. Please continue watching the videos. Thank you.